Hi, now we want to discuss about DNA replication and PCR. So DNA replication is making DNA from a DNA template. So why do we need this? So when the cells divide from one cell to another, so one become two, we need an exact copy of its own genetic material, DNA. So that's when DNA replication is necessary. So think about this double-stranded DNA, and we unzip those, and how we can replicate those. So far, what we know is, and look at this side when we unzip. So this looks like a fork. So that's where we call it a replication fork. And we need an enzyme for producing this. So remember, DNA only makes from 5' to 3' end. So one side, one strand is 5' to 3', then the opposite side should be 3' to 5'. And our creation, making the DNA based on this template, it should start with 5' end to going towards 3' end. So this part is called a leading strand because this polymerization can happen continuously. So what does this job is the enzyme called DNA making it polymer. So we call it DNA polymerase. This catalyzes the reaction that adds a new nucleotide to the growing strand of the DNA, in this case, this. Then thinking about this problem, the opposite side has an issue because look at this DNA, the opposite side is 5' to 3'. Then to make a new copy from 5' towards 3' will be the opposite. While this fork has been elongated, we have only this continuously make it. So for example, like this is 3' end towards 5', then we should be able to have 5' to 3'. And then once this elongates, we need another exposed one. So we have to, again, make from 5' to 3' end. So likewise, when this will unzip, we have to have make it 5' to 3' DNA polymer. But this has to be discrete. So this is discovered by Okazaki, and we call it as Okazaki fragment. And while this is called leading strand, the other side is called a lagging strand. So DNA replication is initiated at sites called replication fork, which is called this look like fork structure. And DNA pore or polymerase requires here a short segment of nucleotide to initiate polymerization. So that we call it as a primer. And this DNA synthesis can only occur in the 5' to 3' direction. The lagging, falling behind, meaning this part strand can only be replicated discontinuously to form Okazaki fragments. So which much then then you have to join this together. So that tie-up is called ligate. So to form a continuous strand so we need an enzyme doing this ligation. So that's called, of course, DNA ligase. So this replication, and there's a number of enzymes involved. You don't have to remember all the enzymes, but pretty basic ones, I want you to understand those. So the double helix is unzipped and unwound. And then each separated strand. Let me show you here. So these blue ones are template, unzipped and unwound. And each separated strand acts as a template. So there's two templates for replicating a new partner strand, which is shown in green here. And look at this direction. So this is a leading strand, so 5' to 3' continuously, while the other side, you have to do it discontinuously, this part and this part. And then while doing this, nucleotide, so these bases are matched A, T, and GC. to synthesize new partner strands into 2 new double helices. So on the other side, look at this structure. This is a leading strand because you can start a primer here, a red one, and 5' towards 3' continuously, while the opposite side, called lagging strand, you can do it during this time discontinuously. So 5' to 3', 5' to 3', and you have to ligate those. So the enzyme machineries, there are a number of enzymes to do this. First, you have to unwound these helix. That's done by topoisomerase, and you have to unzip these DNA double strand. So that's done by helicase. And this part, if you look at closely, so 5' to 3', so the DNA synthesis can start from 5' towards 3' and so continuously. So that this part is leading strand. On the other hand, 3' to 5', then you have to have the opposite side. From here, a short part of primer, you kind of make this DNA and then DNA as Okazaki fragments, and to rejoin them, you need DNA ligase.